I'll introduce this session speaker, Madam Dr. Kalyani Kumari. Presently, she's working as a senior scientist in the discipline of seed science and technology at the National Institute of Seed Science and Technology, Mau, Uttar Pradesh. She completed her BSc from JNKV Jabalpur, that is MT, and followed by MSc and PhD from Anand Agriculture University, Gujarat. She started her career at IRI Regional Station, Pusa, Bihar. There she worked for about 4 years from 2015 to 18. Then she moved to this our National Institute of Seed Science and Technology. Since 2018, she is working in this institute. Madam is having around 10 years of research experience in the field of seed science and technology. She is currently serving as a senior scientist at ICR National Institute of Seed Science and Technology. Her research area focuses on seed physiology and seed testing. Madam, I welcome you for this session. You can start your presentation. Thank you.
Thank you very much. Good afternoon to all. And in coming lecture, we will discuss about seed longevity and deterioration. First of all, regarding seed longevity, a seed's lifespan or longevity is the period over which a seed can remain viable. And the longevity refers to the period from maturation to the loss of seed viability in dry storage conditions. And the longevity of different species and different crop types varies depending upon their chemical composition, their structural features, and storage environment. And on the basis of their storage behavior, we generally classify these seeds into 3 different categories. The first one is orthodox seeds, and these types can generally undergo dehydration before shedding, so they are desiccation tolerant. They can be stored successfully in a dehydrated state at a below freezing temperature for a longer duration. And they can survive a very low moisture content, that is 3 to 5%, because they contain not very 10 to 15% water, less than 10 to 15% water content in their cell structure. And they can tolerate subsequent sub-zero temperatures. And most of the seeds belonging to cereals, pulses, and oil seeds comes under this category only. And the second one is recalcitrant seeds, and these seeds are desiccation intolerant, and they do not survive very below moisture content ranging from between 30 to 65%, depending on the different crop species. And mostly the tropical and subtropical species of fruit crops like lychee and mango comes under this category. And even that cocoa and coffee like beverages seeds also comes under this category. Apart from that, the plantation crop seeds like rubber, oil palm, and coconut seeds also comes under the recalcitrant type of seeds. And apart from that, the many timber species, especially in Dipterocarpaceae family, also belonging to this type of category. And that the temperate species of like oak, chestnut seeds also comes under this category. And they can they are very desiccation intolerant, and we cannot store by reducing their moisture content below 30%. And the third category is intermediate type of storage behavior, and the term itself identifies that they do not behave exactly like orthodox, or we can say recalcitrant. These types of seeds comes show the behavior in between these and above 2 types, and they tolerate a greater degree of dehydration. And the typical recalcitrant seeds, greater degree of dehydration than recalcitrant seeds, and less tolerant to dehydration than orthodox seeds. And when we dry these types of seeds, they lose viability more rapidly to 0 to minus 20 degrees Celsius. And if then at warmer temperature, that is around 15 degrees Celsius temperatures. And the oil palm seeds and some like neem seeds, carica papaya seeds also come under this category only. And this flow chart actually describe to determine the seed storage behavior or longevity pattern of different seeds of any unknown species. And when we get the fresh seed, and we have to determine the water content in that particular seed. And when we go for germination test, And after drying until the 12% moisture content, when we do germination test, if most of the seeds are dead, if they do not germinate, then it comes under recalcitrant category. And if most of the seeds remain viable and we again dry it until 8 to 5% moisture content and again do germination test, then if most of the seed dead, then it also comes under intermediate. And if most of the seeds remain viable, and we can go for 90-day storage at minus 18 degrees Celsius, and by doing germination test, if most of the seeds remain viable, then it comes under orthodox types of seed category. This diagram shows the different cytological cell shrinkage behavior in case of orthodox and recalcitrant seed tissues. We can see that upon maturation and drying, if the cytoplasm, if the seed is undried, then this is a condition of mitochondria and nucleus and vacuoles under the cytoplasm. And when after drying, in case of orthodox type of seed, we can see that they survive mostly when we solidifying, but in type of recalcitrant seed, they do not survive. In case of orthodox seed, we can see the dry mass accumulates during maturation and fills the cell volume. So desiccation forces only slight cell shrinkage. There is a very slight shrinkage in case of orthodox type of seed category, while in case of recalcitrant type, there is accumulation of space filling reserves that is less marked and causing considerable cell shrinkage when drying force is applied in that particular type of seed categories. There are different factors which determine the longevity behavior in different crop species, and the first one is the seed factor. Whatever the type of seed, their composition, chemical composition, their structural features determine their longevity behavior. And we can say that the storage potential of orthodox seed has been attributed to cellular protective mechanism. And at the end of seed development, the orthodox type of seed undergo the maturation process, which corresponding to decline in moisture level and suspension of their metabolic activities as compared to recalcitrant type of seeds. And inside the seed, their chemical composition and other cytological features, we can see that there is the role of different chemical protectants in various tissues. The main damaging factors inside the seed storage is the oxidative stress, which is induced by the reactive oxygen species, reactive oxygen species. And we generally see the molecular enzymatic antioxidants are essential for the duration of longer duration of these types seed longevity. When there is more production of rows, there is decrease in lifespan of that particular type of seed. Apart from that, the role different hormones like abscisic acid, auxin gibberellic acid also play a very important role. We all know that in presence of abscisic acid, seeds force to dormancy behavior, shows dormancy behavior or lack of germination. While there is increase in gibberellic acid, we can see there is profound development of their germination potential behavior. Apart from the seed, inside seed factors, there are different environmental factors belonging to different storage environments. And in that, the moisture content and water activity or equilibrium RH is the most important factor. And we see that in higher temperature and in presence of moisture and oxygen, there is oxidation of lipids, proteins, and nucleic acid inside the tissue and cell of different seed categories. And as we all know that the thumb rule related to the moisture and temperature, Harrington's thumb rule for quantitative effect of humidity on seed longevity or aging, that is if the seed moisture content is between 5% to 14%, each 1% decrease in seed moisture, we can double the shelf life of seeds, lifespan of that particular type of seeds. Apart from moisture and relative humidity, there is also glassy or liquid cytoplasm formation. During the late seed maturation, what happens actually, the drying leads to the transformation of that cytoplasm from fluid state to viscous type, glass type viscosity. That is the form due to the disruption of normal crystal matrices. And that glass phase transition is crucial for the physiological status of that seed, which respond to particular environmental conditions. And again, the temperature is the most important factor. If the temperature is higher, the seed deterioration is greater, and these accelerate the rate of chemical oxidation, which produce the enhancement of rows, and ultimately there is deterioration of that particular type of seed. And again, the second rule, thumb rule given by Harrington's related to temperature is, if the storage life of the is approximately doubles for every 5 degrees Celsius decrease in temperature. And oxygen, as already told, that the Ross formation always decreases the lifespan, longevity of seed. Under dry storage condition, we can see the molecular oxygen is the main source of reactive oxygen species, and that result in the oxidation of lipids, proteins, and nucleic acid. And the seed deterioration under dry condition, if there is a higher oxygen concentration, the faster aging of that particular seed shows their behavior. And low oxygen and anoxia storage under dry condition can extend the seed longevity. Apart from that, the biotic factors, that is pest and pathogens, in the presence of relative humidity and higher temperature and oxygen, the deterioration, they directly affect the seed storage behavior. They play a very important role in the survival and multiplication of pest and pathogens, which impact the loss of viability of that particular seed. And when we go for the period of survival under the natural conditions, on the basis of longevity, we again differentiated the seed into 3 different categories. The first one is macrobiotic. Those types of seeds survive more than 15 years, even over 1,000 years. And they are often hard seeds, having their seed coat is impermeable to water. And many species shows their lifespan up to 55 to 100 or even on 1,000 years. We can see in case of lotus, the seeds are well known for their longevity for even several hundred years. And the second type is mesobiotic, which is their lifespan is mostly 3 to 15 years, and majority of the species comes under this category only. And in this, also there are 2 subcategories. If their lifespan comes under 3 to 5 years, mostly like rapeseed, the pulses, and oilseed crops seeds comes under this category. And if it comes under 5 to 15 years, mostly cereals crops seeds and some vegetable crop seeds comes under this category. And the third category is microbiotic seeds, and it can survive mostly up to 3 years under natural conditions. And most of the recalcitrant seeds and some oligogenous orthodox seeds having more oils and fatty seed compositions exhibits such type of behavior, having short longevity. Mostly vegetable seeds and ornamental crop seeds comes under the microbiotic seeds category. Here, the seed longevity charts of different vegetable and fruits, and we can see the different storage behavior, longevity behavior in different types of seeds. And in most of the vegetables and fruits, we can see the 2 to 4 years or maximum 6 years of lifespan in these categories. While coming to the flower seeds, we can see mostly 1 year or 4 to 5, maximum up to 5 years comes under these types of seeds. And in case of herbs, mostly 1 to 4 years category comes different types of herbs, shows their longevity behavior. Coming to the seed deterioration part, we can say whether the seed is having long lifespan or short lifespan. If there is the natural deterioration process also occur inside the seed. Apart from that, due to different environmental factors and storage behavior, the seed loses their viability or their longevity, and seeds got deteriorated within the span of time. So the seed deterioration is inexorable and irreversible degenerative changes in the overall quality of the seed. If the seed deteriorated, they lost their quality, viability, vigor along with the time due to aging or effect of adverse environmental conditions. And these deteriorative alterations occurring with the time that increases the seed exposure to external challenges and decreases the ability of that seed to survive under stress or different unfavorable environmental conditions. And the rate of deterioration is increases with the increase of seed moisture content and temperature inside the storage conditions. This diagram show the percentage viability and along with the storage time, and we can see that the viability responses in different storage time when this is the when the seed reaches to their threshold level of their aliveness or longevity duration, and after that we can see that there is a rapid mortality behavior with the storage time passes. Apart from that, we can see that when the storage period increases, there is the decrease in the normal seedling percentage in seed, and along with there is abnormal seed increase in abnormal seed, and after that there is declination of abnormal seedling percentage, and there is rapid increase in dead seedling percentage when the seed storage passes after the different storage duration. Apart from that, the abnormal seed abnormal seedling percentage is also decreases, as already told. Dead seed percentage is increases as the storage period passes. This picture also shows the seed longevity behavior along with the storage time, and we can see that the longevity is determined by the genotypic factor and environmental factor inside in the seed, and we can see that as the time possesses the abiotic stresses and different biotic stresses leads to the decrease in viability and longevity of that particular seed type. And due to the oxidative stress, electrolytic leakage, and damage to different cellular constituents inside that seed. And we can see that there are different protectants inside the seed which reduces their deterioration, that is the protected seed coat and antioxidant and reduce metabolic activity. And there is the reparation and degradation renewal, that is protein, DNA and mRNA degradation and lipid carbohydrate degradation and membrane integrated damage, and the detoxification of that is the ROS production and toxic compound, which leads to the decrease in their lifespan. Where does the seed deterioration occur? Where if we talk about the seed deterioration, does it occur uniformly in the whole seed, or there is a different behavior or different pattern for deterioration? Yes, in case of monocot, we can see that the deterioration begins at the root tip, and it causes the radical extension to be reduced more than coleoptile extension. While in case of dicot seeds, we can see there is it begins in the growing points, that is shoot and root of embryonic axis. And there are different types of seed deterioration. The deterioration itself started in certain condition in case of field itself as a form of field weathering due to different unfavorable environmental condition of even higher temperature, higher relative humidity. And apart from that, during harvest and post-harvest deterioration also occur in many types of seed. And apart from these two cases, the most important is storage factor, where the maximum seed deterioration occurs if the temperature and relative humidity increases as the time or storage duration passes, because of the different stress factor developed by the biotic factors, that is the increase in the number of insect pest damage or the different bacteria or virus bacteria and fungi number increases as the temperature and moisture increases, and these all processes deteriorated that particular type of seed lifespan. Again, the characteristics of seed deterioration, as already told, it is an inexorable and irreversible process, and deterioration is the lowest at the time of seed maturation. And we can see the deterioration even varied among different kinds of species. Even among the seed lot of the same kind, if stored under the same condition, we can see the different rate of deterioration in a particular seed lot. And the different deteriorative changes we can see in seeds are it may be changes in the seed coat color or embryo or endosperm color, delay of radical emergence and seedling growth. It may occur in the form of reduction of total germination potential of which leads to their low planting population, an increase in number of abnormal seedlings, and lower tolerance to adverse storage conditions, loss of overall vigor potential of that particular seed, slower rate of seedling growth and development, decrease in germination percentage, stunting growth of radicular plumule, and decreased resistance to environmental stress during germination and early seedling growth. And these all leads to ultimately reduce the yield potential of that particular seed, and complete loss of germinability and death of the seed. Apart from that, the impairment of energy yielding and biosynthetic mechanism also get affected, and reduced biosynthesis and respiration rate, reduction in germination rate and early seedling growth, reduction rate of plant growth and development, and reduced storage potential, and decrease the uniformity in the growth pattern of that particular seed lot. These are the different factors which determine the deteriorative behavior in the seed. In abiotic factors, the temperature, moisture, and the fluctuating environmental conditions also play a very important role. Apart from that, the oxygen pressure, which leads to the formation of more ROS, which ultimately deteriorated the seed quality. And even in biotic factors, the bacteria, fungi, insects, and mites play a major role to deteriorate the quality of seed. Apart from these two, already there are many factors inside a seed which results, which rate is deterioration as the time or storage duration passes. It depends upon the kind or variety of that seed, their genotypic factors, and what is the initial seed quality, and their mode of procurence, and the toxic substances present inside that particular seed lot. This diagram represent the different intrinsic and extrinsic factors which determine the seed deterioration. Under intrinsic factors, we can see the seed moisture content, the type of seed variety and cultivar, storage temperature, and storage hygiene. These are the important factors. And under extrinsic factors, the seed health, what is the seed maturity and composition, and even seed is whether seed is mechanically damaged or not. These also play a very important role. The microbial load, of course, the packaging and storage material, and relative humidity and storage duration. All these play a very important role to the deterioration of that seed quality. Now, what are the biochemical changes which occur during seed deterioration, which leads to the poor quality and decrease the lifespan of seed? The first one is first, when the seed deteriorated, there is the lipid peroxidation occurs, which during lipid peroxidation, it leads to the membrane leakage and loss of integrity of cytoplasmic membrane, which affect the seed viability. When free radicals attacks the unsaturated fatty acid in the cell membrane, which results in the oxidation and degradation of these lipids, and it causes the damage to the cell membrane, and it leads to decrease the membrane integrity and increases the permeability of the cell membrane. If the antioxidant system within the cells help to prevent such type of lipid peroxidation behavior by neutralizing the free radicals and other ROS, reactive oxidation or oxygen species inside the seed tissues. Second one is the membrane degradation. Under the harsh storage condition, there is the degradation in loss in membrane permeability, which leads to the leaching of seed constituents, and which leads to the loss in viability of that seed material. The protein oxidation in the presence of ROS, the attack ROS usually attacks the modify the amino acid sequence residues of proteins, which leads to the degradation of the protein structure and function of proteins, which ultimately reduces the enzymatic behavior and which leads to the protein aggregation and decrease in protein solubility. Again, the changes in the ROS level, when the ROS accumulation increases in the form of hydrogen peroxide, superoxide anions, and hydroxyl radicals, that is increases as the seed ages, and this leads to the oxidative damage of biological macromolecules such as lipids, proteins, and DNA, RNA. We can see that as the degradation of lipid occurs, which leads to the oxidation of vitamin E, lipid peroxidation, and formation of mineraldehyde, hexane protein, and conjugated ions, which leads to the membrane damage. And ultimately leads to the membrane integrity, loss of membrane integrity, and mitochondrial dysfunction. When it deteriorated the protein's quality, it leads to thiol oxidation and carbonylation, which damages the calcium ion behavior and ion transport system, and disruption of ion gradient, and ultimately leads to the deactivation of various enzyme systems. Or these all processes, after that the RNA and DNA damage, which alters the gene expression, ultimately leads to the cell injury and death, and these all behavior leads to the deterioration of that particular seed lot. Again, as the seed deteriorated, there is the changes in the sugar metabolism. The balance between the sugar synthesis and degradation disrupted, which leads to the changes in the sugar metabolism. And the level of the soluble sugars like glucose and fructose decreases during the seed aging, while there is increase in level of oligosaccharides such as raffinose and stachyls. These changes in sugar metabolism is the result of increased activity of enzymes involved in the breakdown of sugars, like alpha amylase and beta glucosidase, there is a decrease of activity of enzymes, those enzymes which are involved in the synthesis of sugars, and the increase in the level of oligosaccharides, which can affect the germination by reducing the water uptake and increase the osmotic potential of the seed. Again, there is a changes in the hormonal levels, like we can see the level of various hormones is changes, such as the abscisic acid, gibberellic acid, and changes as the seed ages. The level of abscisic acid, when that is the stress hormone, play a very important role in seed dormancy and germination, and there is the increase in the level of abscisic acid when the seed ages, and there is the increase in these abscisic acid level leads to the maintenance of seed dormancy, and which can delay or prevent the seed germination. On the contrary of this, when the level of gibberellic acid is increases, it promotes the seed germination and growth, and known to decrease the during seed aging. This decrease of gibberellic acid level can contribute to the loss of seed vigor and poor germination. And apart from these two hormones, also there is a changes in the level of cytokinins, which play a very important role in the cell division differentiation. And we can see there is the decrease in the level of cytokinins as the seed ages, which can affect the germination and growth potential of that seed lot. Apart from that, the alteration in different peroxidase enzyme activity, there is decrease in activities of different peroxidase enzymes as the seed ages. And due to that, the seed becomes more sensitive to the effect of oxygen and free radicals and in membrane, and the unsaturated fatty acid, and which produces the lipid peroxidation and lipid peroxidation products such as MDA and lipid conjugates. And the reduction in metabolic activities of seed is lower in non-viable seeds than in viable seeds, and in the long-term storage decreases the ability to form nucleic acid and nucleotides. While the accumulation of free radicals produced by the different metabolic process leads to deterioration of that particular seed behavior. Also, as the seed deteriorated, there is different chromosomal aberrations is seen in shell tissues. We can see that it may be shows in the form of like fragmentation of chromosomal part, like formation of bridges, fusion, and ring formation in chromosomes, and variation in nuclear size is seen in deteriorated seed. There is degradation of functional structures. There is a lower level of ATP synthesis in deteriorated seed, decline in sugar content, inability of ribosome to dissociate, and enzyme degradation, inactivation, formation and activation of hydrolytic enzymes, starvation of meristematic tissues, and increase in seed leachates and free fatty acid content, and reduction in respiration and accumulation of toxic compounds. This flow chart shows the overall biochemical behavior or deterioration during as the seed ages, and profused different cell behavior in different storage conditions when the seed ages. We can see that when there is the degradation of membrane and degradation of functional structure, which leads to increased concentration of electrolyte leakage, which results in the decline in seed germination, field emergence, and seedling vigor. While when the changes in cell chemical constituents, which leads to the decrease in protein and oil content, and reducing sugar, which increases the free fatty acid, and ultimately result in decline of seed vigor. Enzymatic alteration, low activity of different enzymes, reduced metabolic activity, and free radical damage, which results to the accumulation of free radicals and forming hydrogen peroxides and damages nearby molecules, degradation of food reserves, which is associated with a reduced capacity for protein synthesis and increased amylase synthesis, and decreases lipid content, accumulation of toxic compounds, lipid peroxidation behavior, and different genetic changes, and the mRNA and DNA degradation, and different chromosome aberrations. These all deteriorative behavior ultimately leads to the overall decline in vigor and deterioration of that particular seed lot. This diagram also shows the different deteriorative behavior inside the seed tissues. We can see that there is the lipid, due to the lipid peroxidation, there is the damage of plasma membrane structure, and due to the protein carbonylation, there is abnormal accumulation of protein and enzymatic activity. Due to different decrease in enzymatic activity, you can see the antioxidant system gradually collapsed, and the DNA hypermethylation also occur, and these leads to the structural damages in genetic level. Here, this is the case of in the case study in case of soybean. We can see where the seed is imposed to the accelerated aging for different duration. We can see the trend of changes in the reducing sugar. As the time passes, there is an increase in the level of reducing sugar, and there is a decrease in the concentration of amylase, and there we can see the decrease, the trend behavior in case of catalase enzymes, while there is increase in the electrical conductivity due to the cell membrane disruption behavior as the accelerated aging duration passes. And we can also see there is the decrease in the carbohydrate percentage and the peroxidase behavior trend. We can also see these overall different biochemical changes is seen in case of soybean as we force the seed to accelerated aging by imposing seed to higher temperature and higher relative humidity conditions. This is also a case study in case of sunflower in different seed lot. We can see in case of artificially aged condition of 3 days and 5 days, and in control conditions for 6 months and 12 months, and conventional storage condition under 6 and 12-month condition. We can see the pattern of that lipid peroxidation behavior and super SOD activities, and in different seed lot as the duration passes. There is also as the free fatty acid pattern is increases when the duration of accelerated aging days increase, and there is the decrease in peroxidase activity when we impose the seed to higher number of days while imposing the accelerated aging treatment. This is the case in cotton seed. This is also in case study in case of soybean, and we can see the pattern of different fatty acid content as the storage duration passes. in the 6-month and 12-month in control condition and conventional storage condition, we can see the behavior and trend of changes in the different fatty acids like linoleic acid, oleic and oleic acid, oleic and linoleic acid content. Apart from that, the lipid peroxidation behavior, we can see how the pattern increases and then decreases. And in case of the trend, when the fatty acid content, how that these pattern changes. Now, the how we can control, if there is any remedy, or we control the seed deterioration process. Actually, the seed deterioration is the irreversible process, so we can't stop it. But by doing some conditions or applying some favorable conditions, we can say we can enhance the longevity or storage potential of that particular seed lot by taking some precautions, we can say, or by applying some control measures. Like use of antioxidant, we can protect the free radical formation, and important antioxidant like alpha tocopherol and different ascorbic acid and glutathione. These types of phenolic compounds, by use of that antioxidant, we can protect the formation of free radicals, which ultimately leads to reduction in seed deterioration behavior. Apart from that, we can use the halogen vapors to stabilize the unsaturated fatty acids, which leads to the formation, degradation of cell membrane. By reducing the amount of oxygen, we can increase the initiation of free radical formation, and we can increase the lifespan of that particular seed lot. And there is if the provision of control condition in storage, we can lower down the temperature and relative humidity, so that there is the lesser infestation of insect pest and different mycoflora, which ultimately leads to the greater enhancement of the lifespan of that particular seed. Apart from that, we can also use different seed priming treatment to control the deterioration process, depending upon the type of that crop species. And it has been shown that by applying that aminobenzoic acid and hydrobenzoic acid, we can improve the germination percentage in case of mustard seed. So that's all about that longevity and deterioration behavior of seed. Thank you.
Thank you, madam. Thank you, madam, for nice presentation. Really, it was informative, madam. Then topic is open for discussion. I request participants to interact. Any clarifications, questions, you can raise now.
Very good afternoon, madam.
Good afternoon.
Ma'am, actually, ma'am, we are from Andhra Pradesh state, ma'am. We are doing seed production in coastal area, ma'am. That is the area with high humidity and sometimes prone to high temperatures also. In that situations, what are the precautions we need to take during seed storage and for improving the seed longevity, madam?
Actually, while storing the seed, because you are in the coastal area, automatically there is the more relative humidity in that area. So you have to actually manage the lower down the relative humidity. You can use the different, if the your seed material is in less quantity, you can use the different desiccator cabinet if you are using that seed material for research purpose. If the seed material is in large quantity and in storage conditions of you have to store even quintals of seed, you can use the dehumidifier in your storage, in your storage room or storage godowns. You can use the dehumidifier, so that the relative humidity can be decreased, and it will increase the lifespan of that seed lot.
Madam, we came to know that there are certain types of bags. They can also improve the seed longevity. May I know the details, ma'am?
Ultimately, the major role played by these types of that particular bag, or we can say that precautions, which ultimately results into the lowering down the relative humidity and temperature. If you manage that conditions, you can increase lifespan of